Hi everybody. I see you huddling around the TV in the res. Like to see that happening. Good working together. And I see Michelle and I see Ryan and I see a few others over there. There's Jen. How you doing? Now you might be wondering, what are we doing? Why am I doing this? Well, part of it is that today's class is on sensation, and we're also going to be looking at perception next week. And when we talk about sensing, is we look at all the sensory information that we can take in and how we make sense of it in our world around us, in our brain, how it gets in there. Now, certainly what I sense is I just know people's names, and I have a camera right there, lights, green screen, and of course you see something and you're sensing something completely different than I am. You're seeing a video of me standing in a little white box in the corner of a PowerPoint, and the screen back here isn't green at all, it's a PowerPoint of a chalkboard. So we sense different things in this particular experience, and yet even with the different sensing, we can both perceive the experience pretty similarly. You might know that I use green screen, and so you might be able to imagine what this looks like here. But I know what it looks like from this end, and I'm only imagining what it might look like in the res, or at your home, or in the school where you're sitting and watching this video. So what we're going to be looking at in this week is sensation, just that. The picking up of information from our sensory organs and having it placed, if you will, transferred into our brains, so that when we talk next week, we talk about perception, so that we can take that information and make sense of the world around us, and enabling us to navigate it, to find our way around, to interact with the people in our lives. So that's what this week's on. So my hope is that this week you're going to sense a good time, and next week you perceive it was a good time. Good luck with this week and next, and enjoy the videos. Bye now.
Okay, we're going to look at the definition first of what is sensation and what is perception. And remember, we're just doing sensation this week, but we'll do perception next week. When we look at sensation, we can look at the definition as being a process through which the senses can pick up the visual, the auditory, and any other sensory stimuli and then transmit them to the brain. And what we're really going to do through this particular chapter on sensation is look at the different means by which we do that. And it's quite remarkable the kinds of ways in which we take in information, stimuli from outside, and how we transfer or transmit or change the visual or auditory or feel or smell or taste and translate it into something that the brain can recognize and then sense and organize it and make sense of the world around us. Now, perception by definition, when we look at sensation and perception, so all the sensory-based information is going to help us with our sensation, and perception would be the activities that goes on in the brain. It's the process in which the sensory information that is actively organized and interpreted by the brain. And so what's interesting about that is that we don't all sense exactly the same. And in fact, if we have a hearing impairment or need glasses or we have other forms of disability, or if we have a processing challenge, it makes our sensation or our sensory receptors somewhat compromised. And therefore, if not everyone senses the same thing in the same way, that's one reason why we don't necessarily see the world the same. The other is in the way that we process information. Now, you're seeing examples here of images of the brain in activity, and you're seeing the resting brain at the top, and then you're seeing the brain while walking, seeing, hearing, thinking, and remembering. The color coding, the brighter the color of red or orange or yellow means high activity areas, and blue means more of a less activity areas. And you're seeing with these different activities where the area of concentration, if you will, on the brain is working to perceive the information that's coming in. So you see the seeing is at the back of the brain, thinking is at the front of the fore of the brain, the frontal cortex, prefrontal cortex, and remembering is centered around different areas that it really just depends on what you're remembering. So sensation is the translating, if you will, the receiving of sensory information and bringing it to the brain, and perception is about making sense of what's in the brain and interpreting it. Now, when we look at, sensing, there are what we call absolute thresholds. So if you can imagine there's a lifeguard, on the beach, the absolute threshold is kind of considering what is the dimmest light that the lifeguard could perceive, in the darkness. Like, so what's the darkest it could get, and a lifeguard still be able to do their job successfully. Now, absolute threshold, this measures have been established by sensory psychology. So what you're seeing on the screen would be these different absolute thresholds. These would be the dimmest light or the least amount of taste that can be experienced by human beings in terms of our sensory capacity. Just as a threshold in the doorway is a dividing point between outside and inside of a room, the absolute threshold is a sense marker that marks the difference between not being able to perceive the stimuli and just barely being able to perceive it. So each thing you're seeing here, like for vision, a candle flame from 30 miles away on a clear night, is the absolute threshold. Now, there is such a term as a difference threshold. That's the smallest increase or the smallest decrease required to produce the difference in sensation that is noticeable 50% of the time. So when we think about absolute threshold, and you look at those examples there, you can probably imagine, well, one, maybe some of these I can't do, or people who do these and maybe even better. And so that also explains why we may not all sense the world, therefore perceive the world in exactly the same way. So when we think about sensory information and how we take it in, we are using sensory receptors. And these are highly specialized cells in the sense organs. And you can have specialized vision cells. You have rods and cones. You have hearing cells. You have smell cells, taste, and touch. And all of these are specialized cells for receiving sensory stimulation. And each is sensitive to different sensory stimulation. They detect and respond just to one type of sensory information. Your eyes, cones, and rods only receive information that is a visual of nature. It doesn't receive any hearing or touch. It converts the stimuli into nerve impulses, and this is called transduction. And so transduction is the process that all of our sense organs transmits or converts whatever they receive as stimulation. So for example, in the eyes, they're picking up wavelength, light waves, and the different wavelengths for color. And so your cones will convert those wavelengths into neurological impulses that the brain interprets as color and as images. Now, sensory adaptation is just the ability that based on the frequency maybe that we experience certain sensory information, we can become more accustomed to it. This is the process in which sensory receptors grow accustomed to constant, unchanging levels of stimuli. So if you've worked in a daycare, or if you've been involved in a farm, or if you're like me, I wear shorts all year round, you're basically getting accustomed to different changes in stimuli and become quite comfortable, or at least not as affected as somebody who hasn't grown accustomed. Smokers can grow accustomed to the smell of cigarettes, where they barely notice the smell in their clothes or on their breath, and yet other people, when they walk by them, can smell it easily, particularly if they're non-smokers. And so sensory adaptation is a feature of sensory receptors, that they can get accustomed to different senses. Now, when we think about the eye, we're going to look at a couple of our senses. Visual, we're going to look at hearing, and we're going to look at pain receptors. So when we look at the eye, we have, and I'm going to have you keep an eye on this, be familiar with these different parts of the eye. Now, what you're looking at is light waves will enter in through the cornea, and the pupil will help focus it emits that light and changes in its dilation, and the lens sort of focuses the light that is entering the eye towards the back of the eye, in particular where the cones and rods are. And it's the retina that contains this sensory receptors. Now what you're seeing at the optic nerve is where this sensory information is moved to and is translated or converted into neural information, electrochemical information by which the brain can interpret that visual information. So it used to be that we used to describe that the eyes used to sort of, theoretically would invert images on the back of the eye and the brain would interpret this information. But it's far more specialized than that. It's taking these light waves and converting it into electrical chemical transmissions, neurological transmissions. Okay, now when you think about the light waves, you think, well, geez, it's a whole bunch of stuff. Everywhere you look, there's visual information. And yet, when we think about light waves, we think it's a lot. But if you look at the band across the bottom, where the rainbow is the amount of wavelength that's dedicated just to visual wavelength, the visual light spectrum. On the right, the UV is the ultraviolet rays. Beyond violet is X-rays, gamma rays, and cosmic rays. You'd need different filters on recording devices to pick these up. If they're using telescopes and cameras in space, they're filtering it through filters that allow them to see through or see X-ray, gamma rays, and cosmic rays. And on the other side, we have infrared rays. So if you've been playing with your TV set and you have your controller in your hand and you're changing the channels to your TV with no wires, you're using infrared, and that's a different invisible, longer waves of wavelengths. And the visual part is only this very small band towards the middle of the scale here. And so it's not a very big area that we have visual capability of. Different animals have different access to even UV light ability and even infrared. Now we're going to look at a theory here of color vision. And the trichromatic is one theory that looks at three types of cones, that the vision is using the cones, and there's three types of cones in the retina. Each can make a maximal chemical response to one of three colors, either green, red, or blue. And each one is sensitive to one of the colors. Now if you see the overlap, you can see that you also get yellow and a lighter blue and a magenta, and then of course white. And so trichromatic theory is on this process that the vision is the wavelength is converted by these three different types of cones. And this is what gives us our color vision. Now the alternative view is what's called opponent process theory. And in here there's three kinds of cells that respond by increasing or decreasing their rate of firing when different colors are present. There's red, green, which firing increases when red is present, and green is decreases firing. Yellow and blue, firing increases when yellow is present, and therefore blue is decreased in its firing. And then white and black, firing is increased when white is present and black decreases. And there's an interesting way that you can test this to sort of demonstrate how this actually works. It's called a negative afterimage, and I'm going to show you a picture here. Now what I would like you to do is just pause the video and then expand it to full screen so it's a little easier to do. But what you're going to do is stare at the dot in the green and black yellow flag. Now actually I've changed the flag away from an American flag and put a Canadian flag, so there won't be any yellow in it. But you stare at it at the dot in the middle for a minute, and then you move your gaze to the dot in the blank rectangle you'll see, and then see what you see. There's the green flag, and for 1 minute I want you to stare at that black dot. And then after a minute, stare at the white one and see what happens. Now pause the video and allow yourself a chance to do this, to sort of see the impact of what happens. And I'll share with you later what that is, or why that happens. Go ahead and pause it. Okay, now you might be wondering, how did that happen? If you did this well and you focus strictly on the green flag and on the black dot for 1 minute and then moved over to your vision over to the white and the black dot, you may have seen a black and red Canadian flag. Now there's a reason for that. With opponent process theory, that the red-green cone, while you're staring at the flag, you're fatiguing that cone when it fires green, 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 green. After a while, you fatigue it. Same with the black, 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 black. Then when you move over to the white image, well, the green can't fire off, but the red can because it's not as fatigued, so you see red. The black can't fire off, but the white can, so you see white. And so that's the opponent process theory. Interesting, eh? Okay, let's move on to the ear. Now, hearing, if you will, if we're looking at the human ear, we're doing the same sort of thing as we did with vision. It's coming in with sound waves. And so there's different features. Audation, and the middle ear and the inner ear. Audation is the process of hearing. Auditory, audiation, sorry. The middle ear, this is where it contains the small little bones that connects to the eardrum, to the oval window, and amplifies sound waves. And then the inner ear, the cochlea, this is a fluid-filled, snail-shaped bony chamber, and it contains these very small membranes and hair, cilia, and these hairs are receptors for sound inside the ear. So here's an image of it. Now you're seeing the small semicircular canals where you find these small little bones, the ossicles, small bones named for their shape. There's the hammer, the anvil, and stirrup. And these are the things that vibrate based on the sound waves, amplifying through the eardrum. Now if you see in the small image on the bottom right hand there, you're seeing this eardrum pushing the sound waves, if you will, through the cochlea. And the cochlea is what's filled with these little small cilia. And the little small cilia vibrate. And it's that vibration that is then translated by the brain as sound that we hear. It's an amazing thing what our sense organs can do in terms of how quickly it does it, how rapidly it gets to our brain to help us understand that when we hear that horn blow, we act and react accordingly. I mean, you know, it's amazing how we are put together. All right, so there's a bigger picture of it there. Now, sound waves, they pass through the auditory canal to the eardrum. The eardrum vibrates. The isosceles set is in motion, and in the middle ear, the stirrup pushes an oval window, causing vibrations in the inner ear. Fluid in the cochlea moves back and forth. Hair cells are set in motion, causing a message to be sent to the brain via the auditory nerves. It's quite remarkable how it is that we manage to hear. And then there's theories, of course, for this too. Oh, sorry, we're not going to go to that first. We're going to get back into looking at these different elements about sound. You have frequency, and when you think of frequency, you'll find these in your textbook. Frequency would be the frequency in terms of the wavelength. High frequency is shorter, lower frequency is longer. We have amplitude, which is the height, if you will, the loudness. You'll find these in your textbook. This is a chart looking at decibels. And so this is the sound intensity. And then timbre, which is what separates these, your voice from my voice, is each of our timbre is different, even though we make similar sounds. Okay? So theories for hearing, I'm going to mention just two. There's place theory and frequency theory. Place theory, each individual pitch is determined by a particular spot or place along the basilar membrane of the cochlea that vibrates the most. That's called place theory. There's a particular place. There we go. And so what you're seeing on the left is the cochlear membrane, or the cochlear part of the ear, and on the right you're seeing it lengthened out, and each of these little white indicators, little fingers, are places on the cochlea that would be receptive to different pitches. That's one theory. The other theory is that it's called frequency, where hair cell receptors vibrate the same number of times per second as wave sounds that reach them. So rather than it be a specific place that each type of hair responds or vibrates according to a specific number of times per second frequency of sound. It makes, the art, if you will, of hearing quite a remarkable thing. Now, pain, on the other hand, there's a couple of main theory that's involved with pain, and it's called gate control theory. The area in a spinal cord that acts kind of like a gate. The gate either lets pain messages through or it blocks transmissions to the brain. Slow conducting nerve fibers carry pain messages, and fast conducting nerve fibers carry other messages that can block the pain. This is why sometimes kissing a boo-boo can actually work, is that the fast conducting nerves carry the message of you're being cared of, or while you rub something that's sore, is that it makes it feel like it's good. And it feels good. The slow conducting nerves carry pain. If the fast conducting nerves can get the message to the brain quicker than the slow conducting nerves, then that information is what your brain pays attention to, and that's why sometimes the pain is delayed. Now, there's perceptions of pain that can be influenced by a number of different things. They can be either affected by psychological factors, culture, and endorphins, the body's own natural painkillers are what endorphins are. You can use ice or heat to affect pain positively. And then block pain and produce a feeling of well-being. This is what endorphins do. They're your own little painkillers, if you will. Okay, that's sensation, at least the part that we're going to focus on. Don't forget to use your textbook to sort of highlight some of the key points. Go back over the video if you need to make sure you get some of that information. And anything that I didn't cover very thoroughly, you'll find in your textbook. So I hope you find this useful, and look forward to seeing you in the perception video. Bye for now.